The overall goal of this procedure is to obtain NK or T cell lines from CA-EBV patients, which has great significance of research on this fatal disease.
This method helps to obtain NK or T cell lines for research on the mechanism of CA-EBV, such as studying the role of EBV on cell proliferation, screening inhibitors that interfere in NK T cell proliferation.
The main advantage of this technique is that it is very simple and efficient, which requires only a small amount of peripheral blood and a low dose of IL-2 to establish NK or T cell lines from CBV patients.
Generally, individuals new to this method will struggle because cell survival at the first 2 weeks of culture is uncertain, and this method will show you how to determine cell proliferation at the early culture.
We first had the idea for this method when we found that human serum and IL-2 can maintain human PBMC for about 2 weeks.
Visual demonstration of this method is critical because it shows the shape of the cells and the course of proliferation, other than expansion of cells and numbering the viable cells.
To begin this procedure, purify primary PBMC from 2 ml of whole blood of CA-EBV patients by gradient centrifugation. Alternatively, thaw cryopreserved PBMC from liquid nitrogen with RPMI 1640 medium. Next, add 2 microliters of 0.4% trypan.